Hello everyone, this video is about complement system and this particular study note is developed by EPG Pathshala team, Government of India, MHID. So I would like to express my sincere gratitude for the development team for developing this study note. Thank you so much. Complement system was first discovered in 1890 by Jules Bordet as a heat labile component of serum that complemented or augmented its bactericidal properties. It is known to be comprised of around 30 soluble proteins together, which constitute around 10% of total serum proteins. Most of these proteins are synthesized by liver and remain in inactive form in the absence of infection. Eventually, complement system is an ancient as adaptive immune system, as even worms and starfishes also have complement system. Complement system is the major component of innate system, which is encountered by pathogens that enter our body after breaching physical barriers and antimicrobial defenses. Complement system is the major component of innate system, which is encountered by pathogens that enter our body after reaching physical barriers and antimicrobial defenses. It consists of three different pathways: classical pathway, alternative pathway, and lectin pathway. All these pathways ultimately lead to a common terminal pathway leading to formation of membrane attack complex, which subsequently lays the target cell. Then complement system broadly performs three types of functions: host defense against various infections, interface between innate and adaptive immunity, and clearance of waste. For many of these biological activities of complement system, complement fragments bind various receptors present on different cell types. Additionally, these receptors regulate the complement activity. Besides these receptors, various other regulatory proteins are there that keep a check on the activity of complement system. The importance of complement system in providing immunity to us lies in the fact that a number of diseases result due to genetic or functional diversity of different complement proteins. Still, there is a number of pathogens which have developed evading strategies from complement mediated damage. The mechanism of evasion strategy and the type of microbes possessing these are discussed a later part of this unit. Historical perspective. In 1890s, Jules Bordet at the Pasteur Institute in Paris showed that sheep antiserum to Bacterium vibrio cholerae was lysis of the bacteria. On heating the antiserum, this bacteriophage activity gets lost. On heating the antiserum, this bacteriolytic activity gets lost. But the same can be restored to the heated serum by adding fresh serum. This fresh serum contained no antibodies directed against the bacterium and was unable to kill the bacterium by itself. The fresh serum contained no antibodies directed against the bacterium and was unable to kill the bacterium by itself. So, Bordet reasoned that this bacteriolytic activity utilizes two different substances. First, the specific antibacterial antibodies which survive the heating process, and the second, heat-sensitive component responsible for the lytic activity. Similar experiments were carried out by Paul Ehrlich in Berlin independently, and he defined this phenomenon as the activity of blood serum that completes the action of antibody. He coined the term complement for the same. Later, it was found that complement is inactive composition of large and complex group of proteins. Value addition. Dr. Jules Bordet was honored with the Nobel Prize in Physiology and Medicine 1990 for his discovery of complement-mediated bacteriolysis. Functions. The functions of complement system are broadly categorized as physiological activities of complement system: host defense against infection, opsonization, chemotaxis and activation of leukocytes, lysis of cells and bacteria, interface between innate and adaptive immunity, enhancement of immunologic memory, augmentation of antibody response. Then disposal of waste, clearance of antigen antibody immune complexes, clearance of apoptotic cells. Nomenclature: complement proteins can be named by numerals by C1, C2, C3, C9 up to C9 and like that. Then letter symbols, numerals C1, C2, C3, letter symbols factor B, factor D. Trivial names: decay accelerating factor DAF, homologous restriction factor HRF, homologous restriction factor HRF. The peptide fragments generated upon cleavage or activation of zymogen complement are denoted by small letters. The peptide fragment generated upon cleavage or activation of zymogen complement are denoted by small letters. A smaller fragment resulting from cleavage of a component is designated A, and the larger fragment is designated as B. C3A, C3B. Peptide fragment generated upon cleavage or activation of zymogen complement are denoted by small letters. The smallest fragment resulting from cleavage of a component is designated A, small a, and the larger fragment is designated as B. As exception to this rule, C2 is an exception to this rule, as smaller fragment is called C2B, whereas C2 is the larger cleavage fragment. Just alternate. Okay. The larger fragment bind to the target near the site of activation, and the smaller fragment diffuse from the site and act as a anaphylatoxin. Functional complexes are formed by interaction among complement factors. The complexes having enzymatic activity are designated by a bar over the number of signals. So functional complexes are formed by interaction among complement factors. The complexes having enzymatic activity are designated by a bar over the number of symbols. Pathway. There are three pathways for activation of complement system which culminate to formation of C5b. These pathways are named as classical pathway, alternative pathway, and lectin pathway. The final steps to the formation of membrane attack complex MAC are common for all the pathways, termed as terminal pathway. Then one classical pathway. The classical pathway MAC means membrane attack complex. Okay. The classical pathway of complement activation starts when the complement C1 recognizes a microbial surface directly or indirectly via antibody bound to pathogen. C1 complement is a complex made up of large subunit C1q, which senses the pathogen, and two zymogen subunits sensed by. C1 complement is a complex made up of large subunit C1q and senses the pathogen. The two zymogen subunits C1r and C1s that act as serine protease. One molecule of C1q and two molecules each of C1r and C1s are stabilized together as C1qR2S2 by calcium ions. C1q is a hexamer of trimers. Each monomer of which contain a globular domain at its amino terminal and collagen-like domain on its carboxy terminal. The pathogen recognition activity of C1 lies within the globular domains of C1q. When these globular heads interact with a particular ligand, a conformational change is caused in C1r, C1s, the associated members of C1. It further leads to activation of autocatalytic activity in C1r. The resulting active form of C1r then cleaves C1s to generate active serine protease. C1s acts on two substrates, C4 and C2. C4 is a glycoprotein that consists of three polypeptide genes, alpha, beta, and gamma. C1s hydrolyzes the C4a, small fragment of C4 from amino terminal of alpha polypeptide gene, and exposes a binding site on remaining C4b component. C2 attaches itself to the exposed binding site on C4b and it is cleaved by C1s2, C2a and C2b. The smaller fragment C2b, as mentioned earlier, C2 is the exception in nomenclature, C2b being smaller fragment, diffuses away with C2a attaches itself to target surface along with C4b. The resulting C4b2a complex is called a classical C3 convertase enzyme as it activates C3 from its Imogen form to activate form. Then C3 complement consists of two polypeptide chains alpha and beta. C3 convertase hydrolyzes C3A from amino terminal of alpha chain of C3 and generates C3b. The C3b thus generated binds to C4b2A complex, giving rise to trimolar C4b2A3b complex, which is termed as C5 convertase. The C5 is cleaved by C5 convertase to C5A that diffuses away and C5b which binds to C6 and starts the membrane attack complex formation. Various proteins involved in classical pathway of complement activation are listed. You could see. And comes this picture which can be interpreted like this, leading to formation of C5b, which eventually leads to formation of membrane attack complex C5b. C1QR2S2 complex approaches and gets activated upon binding to antigen antibody complex. C4 is cleaved by activated C1QR2S2 to form C4b, which binds with target surface, whereas C4a diffuses away. C2 is cleaved to C2a and C2b. C2a binds with C2b, forming C4b2a. C3 convertase. C3 is cleavaged by C3 convertase to form C3a and C3b. C3b binds with C4b2a to form C4b2a3b, like C5 convertase. C5 convertase acts upon C5 to form C5b and C5a. C5b initiates transformation of membrane attack complex by terminal pathway. Why circulating IgM does not activate complement system whereas antigen bound IgM does so? Because circulating IgM exists in planar configuration in which CIQ binding sites are not exposed, whereas when pentameric IgM is bound to antigen on its target surface, it occurs staple conformation in which three binding sites for C1q are exposed. Hence, antigen bound IgM can bind to C1q and activate the complement system, whereas circulating IgM cannot. Why IgG is less efficient in activation of classical complement pathway as compared to IgM? C1 molecule must bind by its C1q globular heads to at least two Fc sites of a stable C1 antibody interaction to take place. An IgG molecule contains only one C1q binding site. Thus, for a stable interaction, at least two IgG molecules should be in close vicinity of 30 to 40 nm distance. So, IgM having at least three C1q binding sites binds and activates complement more effectively than IgG. Then alternative pathway. Alternative pathway is antibody independent innate immune system component. It is activated by foreign cell surface and does not require antibody to initiate. In alternative pathway, serum C3 containing an unstable thioester bond undergoes low spontaneous hydrolysis to generate C3A and C3b. The C3b component cannot differentiate host surface from foreign surface, but because of high sialic acid present on host surface, it rapidly deactivates and does not lead to further cascade. Whereas on foreign surfaces such as bacteria, yeast and virus, low sialic acid is present and the C3b remains active. The C3b then binds factor B. So C3b component cannot differentiate host surface from foreign surface, but because of high sialic acid present on host surface, it rapidly deactivates and does not lead to further cascade. Whereas on foreign surface such as bacteria, yeast and virus, low sialic acid is present and thus C3b remain active. So C3b then binds factor B by Mg2 plus dependent bond, which provides a surface for serum protein D to bind. Factor D cleaves factor B, releasing Ba. Remaining complex C3b-Bb possess C3 convertase activity, but it is very unstable. Another factor, Properdin, stabilizes this complex. The C3 convertase further activates C3 to generate more C3b, leading to formation of C5 convertase C3b, Bb, 3b. Then factor D cleaves factor B, releasing Ba. Remaining complex C3b, Bb possess C3 convertase activity, but it is very unstable. Another factor, Properdin, stabilizes this complex. The C3 convertase further activates. C3b then binds to factor B, so there the binding occur. Remaining complex C3b, Bb possess C3 convertase activity, but it is very unstable. Another factor, Properdin, stabilizes this complex. The C3 convertase further activates C3 to generate more C3b, leading to formation of C5 convertase C3b, Bb, 3b. The C5 is cleaved by C5 convertase to C5a that diffuse away, and C5b, which binds to C6 and starts the membrane attack complex formation. And as proteins involved in alternative pathway of complement activation are listed. We learned this. Constituent proteins of alternative pathway of complement activation. Zymogen or native form C3, active form C3b. Function of active form Zymogen, native form C3, active form C3b, binds to pathogen surface, binds to factor B for cleavage by D. C3b-Bb is a C3 convertase and C3b-Bb-3b is a C5 convertase. Then factor B, Ba and Bb not known, Ba activity not known, Bb, which is the active enzyme of C5 and C3 convertase. Then factor D, it is a serine protease that cleaves factor B to Ba and Bb when it is bound to C3b. Then Properdin, P, stabilizes the C3b Bb complex. Okay. Or if the complement activation by alternative pathway leading to formation of C5b, which eventually leads to formation of membrane attack complex. Self and slow activation of small amount of C3 results in C3b formation that binds on an microbial surface. Factor B is cleaved into Bb and Ba in the presence of a factor D. So Bb binds with C3b to form C3 convertase. Properdin stabilizes the C3b, Bb, C3 convertase. C3 is cleaved by C3 convertase, resulting in formation of C5 convertase. C3b, Bb, 3b. Then C5 convertase cleaves C5 to C5b and C5a. Further, C5b participates in membrane attack complex formation by terminal pathway. Lectin pathway. Lectin pathway is also antibody independent. Okay. Innate immune system component. Lectins are the proteins that bind to carbohydrates. This pathway initiates with binding of mannose binding lectin MBL to mannose residues on surface of microorganisms. MBL is an acute phase protein analogous to C1q of classical pathway. MBL leads to binding of MBL associated serine protease MASP, which further cleaves and activates C4. So MASP is structurally similar to C1R and C1S. Rest of the mechanism is similar to classical pathway for generating C3 and C5 convertase, leading to membrane attack complex formation. MBL, mannose binding lectin, MBL bound to mannose, lectin pathway initiated, analogous to C1q. Then MASP, MBL associated serine protease. MASP bound to MBL causes cleavage and activation of C4 analogous to C1R and C1S. Opsonization of pathogen by covalently binding to it facilitates cleavage of C2 by C1S. Inflammatory mediator, cleavage of C1 and C5, C3 and C5, active component of C5 and C3 convertase, vasodilation of C2 kinin precursor, opsonization of pathogen binds C5 for cleavage by C2A, inflammatory mediator. Which one? C3A. Similar kind of representation. Complement activation by lectin pathway leading to formation of C5b, which eventually leads to formation of membrane attack complex. Mannose binding lectin MBL. So, mannose binding lectin MBL binds to mannose present on microbial surface. Activation of MBL associated serine protease MASP takes place. Then MBL activated MASP acts like C1QR2S2 of classical pathway, and rest all the steps are like classical pathway only. Terminal pathway membrane attack complex. This is very important, so learn carefully. C5 convertase upon activation in all the three pathways cleaves C5 into C5A and C5B. C5A diffuses away whereas C5B binds to the target surface. C5B being very labile is stabilized by C6. Upon binding of C7, C5B C67 complex undergoes hydrophilic amphiphilic structural transition, exposing hydrophobic regions that serve as binding sites for membrane phospholipids. If this reaction takes place on target cell surface, C5b67 inserts into phospholipid bilayer, whereas if it occurs on immune complexes, then the released C5b67 complex enters into membrane of nearby cells, causing innocent bystander lysis and autoimmune damage. Next, C8 binds to C5b67, which creates a small pore of 10 angstrom. 10 to 17 molecules of C9 polymerase around C5b678 complex and increase the pore size from 10 angstrom to 70 to 100 angstrom. This pore causes osmotic instability in the target cell because of water influx and loss of electrolytes, ultimately leading to cell lysis. Various proteins involved in terminal pathway of complement activation are listed. Mediates inflammation, initiates assemblage of membrane attack complex, binds to C5b and accepts C7, binds C5b6 and forms amphiphilic complex to insert in lipid bilayer, binds C5b67 and initiates C9 polymerization, polymerize to C5b678 to form a membrane spanning channel that lyses the cell. Different proteins involved in complement system can be broadly categorized in the categories as listed. Binding to AgAb complex and pathogen surface C1q, binding to carbohydrate or pathogen surface MBL, C1q, properdin, ficolins, etc. Activating enzymes CIR, C1R, C1S, C2A, BBD, MS, MASP2. Then membrane binding proteins and opsonins C4b, C3b. Peptide mediators of inflammation C3A, C4A, C5A. Membrane attack complex C5b, C6, C7, C8, C9. Complement receptors CR1, CR2, CR3, CR4. Complement regulators C4b, BP, DAF, MCP, etc. Categories of functional proteins involved in complement system. Parameter precursor C4 and C2 classical pathway. Alternative protease C1S, C3 convertase C4b2A, C5 convertase C4b2A 3B, C5 binding component C3b, activation antibody dependent, component of adaptive immunity. Then alternative pathway similarly C3 and factor B, activating protease factor D, C3 convertase C3b BB. C5 convertase C3b BB 3B. C5 binding component C3b. Antibody dependent activation and component of innate immunity. Alternative pathway innate immunity. So same is the case with lectin pathway. It's also innate immunity mediated. Precursor in lectin pathway C4 and C2. Activating protease MASP. Then C3 convertase C4b2A. C5 convertase C4b2A 3B. C5 binding component 3C C3b. Antibody dependent. First one antibody dependent, others antibody independent activation. Antibody dependent activation, antibody independent activation. Other two alternative pathway, lectin pathway, antibody independent activation. And the classical pathway is a component of adaptive immunity and others are innate immunity. Classical C1 activation upon binding to AG-AB complex. C1 activation. C4 and C3, C4A and C2B, C4B2A, C3 convertase, C3, C4 and C2, C3, C5, C5B. Okay. Alternative slow spontaneous C3B on microbial surface, factor B, factor D, C3, then C5B. Then lectin, mannose binding lectin or MBL binds the microbial surface, MASP, activated C1 like complex C4 and C2, C3, C5, C5B. Then terminal C5B, C5B6, C5B67, C5B678, C5B6789, multiple C9 units, membrane attack complex MAC. And receptors or complement proteins. The biological activity of complement system depends on their binding to various receptors present on different cell types. These receptors not only mediate biological function of complement proteins but also regulate them by inactivating the activated form of complement proteins upon binding. Some of the major receptors and the cells which they are present on along with their ligand and their functions are mentioned. Various cells of receptors for complement components. CR1 complement receptor, CR2 complementary, CR3 complement receptor, CR4 complement receptor, C3A4 AR, like that. Regulation of complement system. Elaborate regulatory system has evolved to restrict the non-specific activity of complement system on host cells. A general way of regulation. Elaborate regulatory system has evolved to restrict the non-specific activity of complement system on host cells. A general way of regulation is achieved by labile nature of all the complement components which undergo spontaneous inactivation on diffusing away from target cells. A number of regulatory proteins also serve as regulators of complement system as listed in table. Many of the proteins are encoded at a single location on chromosome 1 in humans and known as regulators of complement activation RCA gene cluster. The regulatory proteins operate at all types and levels including classical, alternate, lectin and terminal pathway and also at effector level. Then diseases due to complement deficiency. Deficiencies in various complement components result in number of diseases which illustrate the homeostatic roles of complement. Generally, complement deficiencies are rare, but some are very common in specific racial groups. So, some of the common impacts of diseases arising due to complement deficiency are deficiencies of classical pathway. Deficiency of any of the classical pathway component like C1, C3, C4, C2 predispose an individual to a condition similar to systemic lupus erythematosus SLE. In SLE, immune complexes, antigen antibody complexes get deposited in skin, kidney, blood vessels, etc. Then deficiencies of alternative pathway. Deficiency of factor B or factor D reduces the efficiency of opsonization of pathogens. It leads to recurrent bacterial infections in the affected individuals. Then MBL, the lectin pathway deficiency. MBL deficiency in infants makes them susceptible to bacterial infections. But as the age progresses, the susceptibility disappears as other immune mechanisms mature. In adults, MBL deficiency doesn't lead to any serious consequence unless accompanied with other immunosuppression or immunodeficiency. Next, deficiencies of terminal pathway. Deficiency of any of the terminal pathway component C5, C6, C7, C8, C9 predisposes the person to gram-negative bacteria infection, particularly Neisseria genus. So, gonorrhea and meningococcal meningitis are common diseases caused by Neisseria. Then C1 INH deficiency is a classical pathway regulator whose deficiency leads to hereditary angioedema, which is an autosomal dominant disease. Then propyridine deficiency, propyridine, which is stabilizer of alternative pathway C3 convertase, shows X-linked inheritance in its deficiency. This propyridine deficiency is exclusive in males only. Males deficient in propyridine shows meningococcal meningitis. And complement deficiency assay system. Diagnostic approaches for detecting structural and or functional deficiency in complement system are broadly classified as global assays for identification of lack of pathway function, either homeolytic assays or ELISA with IgM, MAN and LPS coated plates is used for such analysis. The total functional activity or functional activity of single complement components can be performed in such assays. Protein chemical, cytofluorometric and functional assay for individual component and regulators irrespective of their functional activity by radial immunodiffusion, nephelometry or ELISA. Molecular analysis to identify genetic alterations, polymorphism in mutation or mutation in complement components can be studied by genetic mapping. Microbial evasion strategies from complement mediated damage. The complement system is very effective in providing immunity against various pathogens, but microbes have also evolved a number of strategies to breach the system. Complement system is most effective against gram-negative bacteria as compared to gram-positive ones. A few gram-negative and most gram-positive bacteria adopt different survival strategies for fooling our complement system. Some of their common evading mechanisms, along with the examples I mentioned: prevention to MAC to insert in bacterial membrane, inactivation of anaphylatoxin C3A and C5A, prevention of MAC insertion, physical barrier between C3b and CR1, inhibition of complement cascade, polysaccharide chains in cell wall, other outer membrane proteins, elastase enzyme, bacterial capsule, peptidoglycan in cell wall, molecular mimics of complement regulators, pathogenic forms of E. coli, Salmonella, pathogenic form of Neisseria gonorrhoeae, Pseudomonas aeruginosa, Streptococcus pneumoniae, Streptococcus herpes virus, vaccinia, etc. Then summary. Complement system is a group of around 30 soluble proteins synthesized by liver and present in inactive form in serum in the absence of infection. They were first discovered by Jules Bordet as an heat labile component of serum that complemented or augmented its bactericidal properties. Complement system is the major component of innate system, which is encountered by pathogens that enter our body after breaching physical barriers and antimicrobial defenses. Moreover, it provides a link in between two different branches of immunology, innate and adaptive. There are three different pathways for activation of complement: classical pathway, alternative pathway, and lectin pathway. All these pathways ultimately lead to a common terminal pathway, leading to formation of membrane attack complex, which subsequently lyses the target cell. Three broad categories of functions that complement system performs are host defense against various infections, interface between innate and adaptive immunity, and clearance of waste. The biological activities of complement system are governed upon binding of complement components and fragments and with various receptors present on different cell types. Additionally, these receptors regulate the complement activity. Various regulatory proteins are there that keep a check on the activity of complement system to avoid the damage to host itself. Then genetic or functional deficiency of different complement proteins result in number of diseases which illustrate the homeostatic role of complement system. Different diagnostic methods are available now to detect the overall complement deficiency or efficiency of any specific component. A number of pathogens have evolved evading strategies from complement system. Particularly gram-positive bacteria and some gram-negative bacteria and viruses are among all these smart evaders.